Hi everyone, my name is Eric Chung, and I, together with Dr. Ullah and Dr. Baranova, are excited to present our recent findings on the topic elucidating COVID-19 pathophysiology in blood, OXPHOS gene signatures in recovered patients. Our study investigated the long-term effects of COVID-19 in recovered patients at 12 to 24 weeks post-infection. This is crucial because despite clearing the virus, some patients continue to experience long-term symptoms. For our study, we utilized the geodata set GXC 169687. The first key step involved differential gene expression, and then the next step, we compared the different patient groups at each time point to the other to identify common gene sets across all the time points. We then teased out the hub gene signature using PPI maps. Our results are shown in figures 1 and 2. For figure 1, A to F, as you can see, there is a dynamic gene expression pattern that starts as mostly downregulation at 12 weeks post-infection, but then switches to mostly upregulation at 16 weeks post-infection, and also mostly upregulation at 24 weeks post-infection. For figure 2 here, figure 2, as you can see, A and B, the results show the final step in our process, which is basically from the initial list of 473 genes, we screen out these genes, have signature. A breakdown of these genes, or a more detailed picture of these genes in terms of how they differ across the 3 time points, is shown in figure C. As you can see, most of these genes are OXPHOS genes, for example, COX7A2, ATP5PO, ATP5F1C, and many others. As you can see, these genes were mostly found to be upregulated at peak levels at 12 weeks post-infection and gradually reduce in expression from 16 weeks post-infection to 24 weeks post-infection. We believe the observed upregulation in OXPHOS genes is a host adaptive mechanism to make up for energy gaps triggered by the virus-induced mitochondrial dysfunction, which happens during active infection. We also observed a sustained upregulation of key ubiquitination genes such as UBE, 3A, and FAU. Our discussion and conclusion here sheds light on the potential of these findings for clinical applications, particularly in improving post-recovery treatment and rehabilitation strategies for long COVID patients. In conclusion, our study provides a foundational step towards understanding the persistent molecular aftermath of COVID-19 and opens up new avenues for future research on treatment strategies for the chronic effects of this disease. Thank you for your attention, and we welcome any questions or discussions on this presentation.